It is now my great pleasure to introduce to you another longtime friend of COFES, Omid Moghadam. And Omid, I think we met when he was at Intel. He is now at Harvard. And I asked him how he would like to be introduced. What are the favorite things that he likes to hear said about him? And he said that he pardon? Handsome. Yes, besides handsome. And he said he received one of the awards when in a meeting that included a bunch of other people. Andy Grove said to him, "You're not as stupid as you look."
And believe me, that's a compliment.
And Omid is with Harvard Medical School, and he's going to speak to us on the topic that is not directly a CAD-CAM topic, and which is what we love to hear at COFES, because we love to bring in the things that are slightly outside of the stuff we deal with on a daily basis to help us get a better understanding of what in the world it is we were supposed to be doing in the first place. So please give a warm welcome to Omid Moghadam.
As Joel mentioned, I used to attend COFES, and for a while I used to run advanced product development at Intel, and trying to teach Intel to pay attention to things other than just spreadsheets and Microsoft Office. And that brought me to COFES, and I learned and I learned to admire the engineering software world. And I tried to imagine a world where if we did not have engineering software, if we had to actually build everything before we could test it, like the old days, when being a test pilot was actually a dangerous job, because you were probably the first one that was testing, validating the design of the airplane. And when I went back to healthcare, which is where I came from, actually, I took a lot of learnings from engineering software back with me. The whole idea of being able to simulate things before we actually build them, and looking at how we can do this in development of new therapies, for example, and new pharmaceuticals. And that's sort of like the general topic that I want to talk to you about today. So it is nothing new to you. You already know that you can design things and build things and validate them before actually even making one of them. But the way we design pharmaceuticals and build pharmaceuticals today is a very 19th century model. It's trial and error. We basically pick any compound that we have, and then they throw it against any disease that we have, and hopefully one of them will stick. And it's amazing that it has worked so well so far, but for some reason it has. But it is running its course, and it's coming to an end. The blockbuster design and the blockbuster drug design and discovery method is coming to an end. It really is not sustainable. But there are better ways of dealing with this. There's a whole new slew of data that's becoming available to us now through genomics and models of protein behavior that we're understanding, and of course abundant and inexpensive computing power that's available to us now, which wasn't in the past, where we can actually design things. We're starting to learn how human body and human body behaves as a system, and how its behavior is regulated, behavior of cells is regulated by things like the genomes and proteins and receptors and so on and so forth. But what is lacking in this area is the right tools. We just do not have the right tools to simulate system biology. So I should relearn biology. I apologize. I had to learn it myself, so I figured I can put one slide together from high school biology. You know, each of our cells has the 46 coiled blobby-looking things called chromosomes that comprise the DNA molecule. This is the long chain double helix the form that everyone's familiar with, and it has 3 million base pairs. Base pairs are those molecules. Thankfully, we only have 4 of them that they go by their acronym. We usually refer to them by their acronyms of CA, G, and T. And these things are arrayed across from one another in 3 billion pairs for the human DNA. And the patterns that the CAGT determine are what's called genes. And some of these patterns are useless, they don't do anything, and some of them actually do something. They regulate creation of proteins and traits that we have, or diseases that we sort of have propensity to get other than the rest of the population, and so on and so forth. So those proteins that are created with this code basically run everything in our body. And that's pretty much as much as I know, so you have the same amount of information that I have. Let's jump into genomics. Where all of this started was the Human Genome Project. It started quite time before that, but we got serious about it with the Human Genome Project. It started in 1990. It was a 13-year project, cost about $3 billion, and this was government funded by the US government. And we sequenced the 3 billion base pairs of one person's DNA, and we identified 25,000 genes through that. And tell you a little bit of a soap opera story. There was actually another effort, a non-government effort by a gentleman called Craig Bednar. And he did the same thing, only he did it through predictive modeling. They didn't actually sequence the whole thing because it took a long time with the machines back there to sequence things. They did predictive modeling. And they did it with $100 million. And then the two groups went into the scientific equivalent of a cat fight for about 10 years to decide who was right. And after 10 years, they decided that they were both right, and they shook hands and they walked out, and they still hate each other. And yes, it's all about science, of course. So we cracked the code of life. And all the business press told us that this is the end of disease as we know it, and we're all going to live to be 1,000, and so on and so forth. So human behavior kicked in. Right after the dot-com bomb, the money had to go somewhere. So in 2003, we started climbing the summit of the peak of inflated expectations of genomics. And money poured in. Money poured in, and everyone was looking for the low-hanging fruit. What is it that we can do with this new information that we have? I need not remind you what happened to the earliest low-hanging fruit pickers. And things haven't changed since back then. So the irrational exuberance of the genomics age actually started way before we actually had the first DNA. We had bits and pieces of it, so people started trying to exploit that and try to use that information for development of therapies. And this is just a random picking of some of the bigger failures. Interleukin genomics actually started to develop cancer drugs, and they're still in business, but the only thing they sell now is a direct-to-consumer test for weight loss, which is very, very sort of shady. It's not based on science. It's sort of like, if you remember a few years back, there was a diet based on your blood type. It has the same scientific value as that. GenSet was founded in '89. Again, they went after cancer, and they have disappeared. I have no idea what happened to them. Insight Pharmaceuticals, after about 10 years of spending on the average $150 million a year on R&D, they were just shut down. Human Genome Sciences, still around. They were founded in '92. They still have no drugs on the market. Varigenics, no products. Sangamo, no products, although they're still in business. Millennium Pharmaceuticals, which was such a spectacular failure that had its own Harvard Business Review case, was founded in '97, and this was the equivalent of, like Enron of this business. And after burning through about $40 billion, I think they were sold for $150 million last year to a Japanese pharmaceutical company. And nutrigenomics, and haven't heard of them, no products. They say their business is on hold. I have no idea what that really means. And most of this was because it was mainly due to availability of data. I mean, don't forget, many of these companies started before we even had the first genome sequenced, and some of them were founded after we only had one genome sequenced. It's like, you know, someone modeling the Wright brothers plane and then taking that and trying to build a 747 based on that information. But that's what happened. So by 2007, you know, we had taken a nosedive off of that summit, and no one wanted to hear anything about genomics, and no investor wanted to know anything about genetics, genomics, you know, sort of like get out, don't let the door hit you in the ass when you asked for investments in this area. The market completely dried up. But thankfully, not everyone gave up. So why? Why was this? What was the problem? Why is it that that now that we cracked the code of life, for God's sake, why is it that we cannot do anything with it? For one thing, this stuff is very complicated. 3 million base pairs. Then we have variants in every human from the other one. And then linkages between those and, say, diseases. And it's not just your genome. Genome is, you know, your genome sets up the pins. Environment and your behavior is really what sort of knocks the pins down, to use a bowling analogy. This is a snapshot of a typical genome-wide association study. That's when you do a study between trying to figure out what common variant within a person's genome can lead to a propensity, for example, to have a higher percentage of chance of getting actually a disease. And each of these things is one genome, and each of these is one person. And then you're trying to find linkages between these things and have a closer look. As you can see, you know, for example, you know, certain genomes have relationship to certain parts of the body, and trying to figure out what it is within this noisy data. It is a complicated task, but it's nothing that we cannot do. For one thing, we do have inexpensive computing, and we can have plenty of it. The other reason why this stuff is so complicated is because there are no standards. You know, the way typically this work is done is that you do one of these genome-wide association studies. You publish a paper. The paper goes through a rigorous process of peer review, and it gets published in a scientific journal. This is actually a real study, and a lot of the information is taken out because I just wanted to show you what happens where there are no standards in measuring these things. These are 13 studies on the same genome, and the circles that you see there is the relative, the conclusion that they reached in each of these studies on whether this gene is protective against a person getting a certain disease or not. And as you can see, it's all over the map. We don't really know whether this is protective or not. Mostly, 11 of the studies said it is protective. 2 of them said it's not. And if you can imagine, if we have only taken a sample of these things, we might have reached the wrong conclusion. And reaching a wrong conclusion is, can be problematic, because this is not just an academic exercise. This is making it into clinical practice. Today there are about 500 single gene tests that people are using, and they make important decisions based on those. BRCA1 is probably the most famous gene, for breast cancer, and other genes, other companion test genes for taking certain type of drugs, for example, like warfarin or Herceptin, which is a blood thinner and a breast cancer drug. So people are making actually real decisions based on these, and since there are no standards on how we score these studies, it makes matters more complicated. So now that I gave you all the bad news, there is some good news too in this area. So in 2007, when everyone else gave up on making drugs based on these things, not everyone gave up on making new tools and new devices. And as a result of that, a whole new generation of gene sequencer machines have come on the market, starting in late 2008, and they have basically set the rate of discovery on fire, not just accelerating. We have increased the rate of data generation exponentially. And as a result of that, there's a lot of public money that's actually going into this area of research that's fueling its growth. NIH has never had a down year. Their budget has kept going up, and more of the NIH funding is going into genomics research. And they just got a one-time boost of $4 billion last year just for more genomics research. And it's not just in the US. There's huge pockets of activity in other places as well. Just this rush to sequence as many genomes as you can, because we understand that really with one we can only learn so much. And these genome-wide association studies have grown amazingly in size. In early 2009, there were only 5 academic research centers around the world that could do these. And right now, I think there are over 200. And probably today, they're going to go up to 210. Because people are just building gene research and genomics research at a fast pace. And the cost of doing sequencing has come down by a factor of 3 million to 1 in less than a decade. I don't think that there's any other area of science that we can actually point to that we've had this level of cost decrease in such a short time. Of course, no discussion of computing is relevant without actually showing, a chart of Moore's law. But actually, I'm showing this for another reason. I'm not going to pile up accolades for Intel for doing this, but for the fact that computing is becoming a bottleneck now in life sciences. Moore's law cannot keep up. In late 2008, the rate of doubling of the rate that we were generating genomics data was every 14 months. So even Moore's law was behind back then. Right now, it's every 6 months. By the end of this year, it's going to be in a matter of weeks. So a lot of companies that are working in this area are looking for alternative ways of doing their computing. They're looking at GPUs. Hopefully, Intel would come up with that Larrabee chip finally. Something that was devised at COFES at one point. So we have abundant computing, even though it's becoming a bottleneck. And also the low cost of sequencing. So this chart shows basically where we have gone from 1990, which was the start, to end of 2010. And we've gone from $10 per base pair. So for a gene, if we have 3 billion of them, 30 billion to about $1,000 at the bottom, or $3,000 at the bottom. And there are new machines that are coming online by the end of this year that's going to bring it down to below 1,000. And by 2011, they're going to be in the hundreds of dollars. So it's going to be cheaper than, or as expensive as a blood panel, for example, a cholesterol test. And that's for the whole genome. So what does all this mean to the pharmaceutical R&D and the so-called system biology area? Just going through the way we devise drugs today, the so-called blockbuster drug model. What is the discovery methodology of that? They start with compounds. They start with tens of thousands of compounds, typically 10,000. That's the cutoff. And through brute force, wet biology, lab testing, they narrow it down to about 1,000. So once we've gone through the test tubes, then we test about 1,000 of them on live cells. So that's called the in vitro trial. And out of those, 20 are taken for in vivo trials, which means animal testing. And of those, if you're lucky, I mean, at any point you can fail in any of these and you have to start all over again. But if you're lucky and you actually find something in those 20, 10 of those compounds make it into the human trials. And there's the human clinical trials. And if you're successful, after about 15 years and $230 million cost, you have a blockbuster drug that costs billions, that would make you billions of dollars. But most of the time it doesn't. The rate is probably 20 to 1 in failure and success. And it's not amazing that this works. I mean, it's not amazing that it doesn't work. It's amazing that it has worked so well so far. And part of it is because of the quirk of the U.S. healthcare system. It's because the cost really does not matter. It does matter in the long run. It does matter when politicians start talking about it. But in reality, people would pay anything because it's not coming out of their pockets. Insurance is paying for it. So somehow this whole idea of maybe cutting time and cutting cost and all of those has never entered this business. And the blockbuster drugs, they have to go for the largest portion of the population as they can. So they look at two things. It should work for a large portion of the population and it should not kill you. So toxic and works, and then you go through this 15-year trying to figure out what works within the trial and error system. And the result most of the time is the opposite of eureka, as you can imagine. So, I mean, from early days of the Human Genome Project, we always thought that molecular medicine is going to be the answer to give us a shortcut through this process. And it can. I mean, genomics data can lead to shortcuts in the process because we can model the behavior of proteins and molecules. And if we can mathematically model these things, we can actually cut the 10,000 to something else. And a lot of people tried that. Not all early innovators succeed in this space. Remember the low-hanging fruit pickers. Millennium Pharmaceuticals was a perfect example. And of all of those companies, it was the very first one, and the only one even since then, that talked about mathematically modeling things. They never talked about pharmaceutical development. They talked about mathematical modeling of system biology. And it had a lot of promise. I mean, they raised tens of billions in their IPO, and they raised more in contracts that they got from pharmaceutical companies to co-develop drugs with them. But at the end of the day, they lacked actionable data. They had one genome, and that was it. And as a result, it was a spectacular failure. I actually met the founder, and he admitted that it was the lack of that actionable data, lack of data that really did him in. And I also checked with him to see if it's true that he showed up to a shareholder conference dressed as a clown, and he did say that he did that actually. But we are learning and we're moving forward from all those failures. There are two problems with this area. The easy one is science. Really, we cannot develop predictive models without enough data, and we're solving that problem on a daily basis now. And on the other hand, our tool development needs to catch up. And even when I was coming to COFIS and I was at Intel, I tried very hard to find a startup in this space that Intel could invest in, that perhaps we can make him into the next Dassault systems of pharmaceutical development in tools, but no one was interested. The other problem, the harder problem as a result, was business. Pharma firms are run by biochemists. So the real men in those companies are biochemists, not computer programmers, not engineers. There are not enough engineers in those places. And if you think that's not a problem, think of Kodak. Kodak developed and invented the first digital camera, but now it owns about 6% of that market. It was a company run by chemists. There are no incentives for development of tools. There's plenty of incentives for development of compounds, but not tools. And there's really no incentive with the healthcare system that we have today. There's no incentive for reducing costs or improving their processes internally, because they always would recoup. Pharma companies are not hurting. And as a matter of fact, this system was really designed to fight or it was designed for the 19th century. It was designed to get us beyond infectious diseases that a large portion of the population have. We don't have that today. I mean, it worked so well that we do not have that problem today. We're gone to chronic diseases, and blockbuster drug model for chronic diseases just does not work. If there is any software tools developed, they are a strategic advantage, not the goal of the company. So if you have a good tool, you keep it to yourself, you develop a drug, you have an IPO or you get sold, and you never speak of it ever again. And probably no one would ever touch that tool again. So what is the opportunity that I see for software engineering firms or any software firm that's interested in this space? If we look at the three market phases of genomics and how it's transforming life sciences, the very early phase, which took about 25 years, was sensor-driven. We really needed to figure out the device that could actually give us the data that we need. And it took a good 25 years in development of those tools. And the biggest chunk of this was really the early development, and it's really only in the last year that we have had devices that can give us real data and real large amounts of data that we can use. And what we worked on in phase one was really digitization of all that data, trying to figure out how many people we can, if we can actually even sequence one person for that database, and visualization of that data. I mean, data visualization was actually really behind in that space, but now that we have caught on. Phase two is really information driven, and that's where we are today. And it's amazing. If phase one was 25 years, we have gone through phase two in less than 18 months. That now we're building very large aggregated databases of human genomes that are publicly available. And we're cross-sampling and data mining these different databases. I mean, there's a very large database on cancers. There's a very large database on humans, 1,000. Their goal is to have 1,000 genomes in there by the end of the year. And we're actually looking for surveillance. We're looking for what's called common variants and rare variants, which are sort of like common diseases and rare diseases. And through those, we're doing what's called network discovery, that we're figuring out what is common between different humans and what is not, and what can lead to diseases and what cannot, and which portions of the genome are useless and which are not. And we really need to get to the simulation-driven part of the nirvana of this space with the promise that they've always been given to us. It's really the model-based biocomputing. And I think that you recognize really from, if you look at manufacturing and engineering software, these are the three market phases that space has gone through as well. And really, this is where we are in manufacturing today. This is where we need to get in life sciences as well. We need to develop the tools that can actually give us the analytic capability to do system biology, to do some multi-scale simulation, and eventually to develop synthetic biology that can fix some of the problems that we have. Is that a sign for us or no? Okay. So we need design tools. And that's where companies like yours come in and knowledge like yours come in. I put the three market phases together just so how it goes. It's not to scale because as I told you, the first phase was 25 years, the second only 18 months. And that's an amazing acceleration. So we need to redesign the process. Going from blockbuster, which was for a very large number of the population, we need to model the behavior to the individual level. A term, personalized medicine, has been thrown around a lot lately, or targeted medicine. But the basic idea is that you don't actually develop a therapy for 80% of the population. You might want to develop one for 10% of the population. And I was reading a report yesterday that some of the studies show that really half of all pharmaceuticals that we pay for or are prescribed to us are useless because based on our genetic makeup, they don't work for us. But under the blockbuster model, we're all going to get those. We're all going to get those, and we take them, and they just push up the costs of healthcare for us. Secondly, we need to develop and simulate tests on a personal level, and we're getting there. By next year, it's going to be in the hundreds of dollars, so we would be able to have these tests on an individual level. And we can assess toxicity and efficacy. Remember, not kill you, be good for you, on the individual level. And there is a very large database of failed compounds out there that we can draw from, so we don't have to start from scratch. But I don't think the industry itself can fix itself. We're going to need new entrants into this field. We need new thinking from outside pharma industry, someone who has a different business model, the business model that they have not had to date. We need experience in design, simulation, predictive modeling, and such. And we need new business models. Pharma does not have this business model, and they're not going to get it unless it comes from the outside. And it's not like it's the first time that this has happened. There's a big push now within the US to digitize our healthcare system, so electronic medical records are becoming big. And some of the best entrants into the EMR are not coming from other EMR or companies who have been in the EMR business for a long time. Some of the most innovative products are actually coming out of the supply chain management industry. And I'm hoping that we can see new entrants into the drug discovery and simulation and predictive modeling of life sciences from outside as well. So why engineering software firms? There is an expertise in development of software for designing, simulation, collaboration, and modeling, exactly what life sciences and drug development needs. Expertise in the business of software, and that's very important, because if you have built your business on biochemistry and wet biology, you're not going to know how to run a software business. So you're going to need those tools from the outside the business to come in. And really, there's no legacy business models to compete with. And that's where the example of Kodak comes in. In conclusion, we have gone through the trough of disillusionment, that was 2007. I think that right now, with the new tools that we have developed and the fact that we're getting so much more data at such a high rate, and the fact that there is so much good information that's coming out of R&D is putting us on the slope. And we need to get to the plateau of productivity, and we can get there fairly quickly, but we're going to need help from outside. So key points, the key points that I wanted to bring to your attention is that the genetic data and design and simulation tools can transform medicine. This is a medicine that was, this drug discovery model and our medical system was built for fighting infectious diseases in the 19th century. It's not suited for what we need today. The current discovery methods are not sustainable. As you know, any analyst that you read, any report that you read, says that the pipelines of large pharma companies are drying up. They just do not have enough compounds in there to sustain their size and their share value. Drug design and simulation must step up to take some of the load off current methods. So that idea of personalized medicine or targeted medicine or making medicines faster and cheaper for a smaller portion of the population is one way of going about that. But to do that, we're going to need the tools. We don't have the tools that are needed for this 21st century healthcare. And the pharma business models are not suited for development of these things. So we need someone from the outside to come and do it. And there is some money to be made here. And I have some further reading if you want to take a look at some of the tools available. There is I2B2 is a project run out of Harvard, and the idea is it's informatics from biology to the bedside. It's for genome-wide association studies. It's a tool that we're developing in-house. The informatics program at Children's Hospital Boston is linking EMRs to genetic studies, and Ignite Institute for Personalized Medicine is an institute that I helped set up, and its goal is to do research just on the personalized medicine. So it's linking of a person's genome to development of therapies. And there's a Harvard Business Review case on Millennium Pharmaceuticals, which makes great reading. for anyone interested. It's full of really good information and a lot of anecdotal stuff. I'll take questions if you have a few minutes. I think you were first. Sorry.
When you're faced with trillion-linked combinatorics, computers become involved because they're designed to do it. So you can see the mind is going to relate other than long-running machines as well.
Yes. Oh yeah. I think the question was that that computing is a bottleneck because they're designed to do arithmetic, and whether there are other machines that we can rely on for doing this work. And absolutely. Actually, the last business that I started for Intel, which they shut down, was basically focusing on that. And the idea was that since Moore's law cannot keep up with this, we need to actually go back to specialized circuitry and sort of like a service model. That it was a mini sort of supercomputer that we put on the side, and we would feed data to it. We feed the latest algorithms to it monthly as a service, and that resonated very well with the industry, but not with management at Intel. But other people are picking that up. I mean, there is a business in that. Oh, sorry. Yes.
There's a number of informational genetic databases such being launched now.
Mm-hmm.
Do they have a standard means of sharing information between them, or means that we write software against that provide
good question. The question was about standards, whether there are any standards between all these different databases. The answer is no. There is no standards, but people are working on something. There's actually two efforts. One of them through the 1,000 Genome Project, which is run out of NIH, and the other one is a private effort that has, well, private-public effort called CDISC, CDISC, and CDISC was actually established for standards in communicating in between different pharma companies, but somehow genomics has gone in there as well. They have a genomics working group as well. Is someone? Yes.
What ideas do you have about writing the software or just making money at
Oh, sorry. What are the ideas around making a business out of these tools? All of these companies are going to need something to shorten their discovery time, and they also need guidance on how they can make it more personal, going from the blockbuster model to more of a personalized medicine model. And I think that's the opportunity. That's sort of like the window of opportunity for developing tools that would teach them how to do that. So it's an enterprise software type business that would outsource that portion of their work to someone else because they just don't have that type of expertise in house. Sorry.
You said that around 2035 money went into the field, and most of it got burned on the way up. If people in our industry were to come up with an idea, is there venture capital ready to invest in the field?
There is. Actually, this area is number one in terms of investment from venture capital. And there's more to it, and there's a lot of public money that goes into it as well. So a lot of these companies in early stages actually they live on grants, they don't live on venture capital. Yes. Hey, John. No. Not that I know of. Oh, I'm sorry. What was that? The following? Folding at home. Thank you. The protein folding, like the SETI at home that you use. that you use the nodes on a network on people's machines to do this. Not that I know of. No, we haven't looked at that. That's a separate effort. That doesn't mean that it's not having that, but it's not, I haven't seen it. Yes. No, it's not. The question was that there is bioinformatics and bioengineering programs at universities. I think what's happening is that a lot of that information never makes it out of universities. And there's also a disincentive with the university work. The money comes from public sources, so everything's thrown into open source. And it has somewhat forgotten when it gets to open source. Actually, part of the business that we were working on at Intel was taking that open source software and actually making it useful to people. True. There's a lot of small companies that take that open source software and then become consulting firms. They don't become, they don't sell the software. They just go and implement it on site. Yes.
A lot of your talks talked about DNA technology and data synthesis, but in order for the correct data to be implemented, a lot of personalized methods require that genetics from proteins. How much new discovery do you think needs to be done in order to be accurate on proteins?
Quite a bit, and that work is going on now. It is separate from the genomic studies, which is linkages between genes and diseases. But the system biology work is also happening in parallel. And the folding at home is one example of sort of like bringing the public into that. But until we have enough, these things can take place in parallel, but they cannot start at the same time. You need enough information on the genomic side before you can actually get to the protein behavior modeling. Yeah.
Several questions. With CAD/CAM we had a century of unchanged geometry, physics, and math. So it was a science of the other than the represent radioactive things. What was that? I think the government did that very well. There was enough science there to actually do the beyond the
We are ways away from actually having a complete system model. Let me repeat the question. That is there enough science behind it before we can do this? We're years away from actually modeling the whole body as a system, but there are subsets of the human body that we can model. So there is science for subsets of it, and where the main focus is right now is in cancer, which is actually one of the most challenging ones. But there is enough information in there, and once we actually have all the cancer tumors sequenced, which we're doing right now, we would be able to actually model that. So there is enough science in some areas to be able to do that, and we have to start somewhere. Okay, there was enough. Yes.
Would you like to comment on the transfer of companies actually
So that's interesting, because there was a ruling out of New York about 2 weeks ago that, the question, would I like to comment on gene patents? And actually one of those genes was actually in my presentation, BRCA1. So there's a company that has patented BRCA1 and BRCA2 genes. And these are the definitive breast cancer genes. And they're charging something like $7,500 per test. And a court, an appeals court in New York, I think, state court in New York, invalidated that patent. Interesting, because it's out of the jurisdiction of that court to do that. So this is going to go to the Court of Appeals for the Federal Circuit, the patent court in D.C. for decision. And it's probably going to be upheld. But what's happening I actually gave evidence to the Parliament of Australia last year against these patent genes, because at the time the government of Australia was actually thinking about invalidating the patents in Australia, which goes against like 80 years of international treaties, that they were going to invalidate the patent that was offered in the US for the exact same reason, because they thought that the royalties were unreasonable. I think that the US Patent Office has become a registration office. It doesn't actually look at the patentability of many of these inventions, and just they kick the can down the road for the courts to decide. And it takes about 10 years. For anytime we have a new area of science or technology, they grant patents. Not too long ago, they were granting patents for methods of doing business or for software patents. And it took about 10 years of litigation to figure out exactly what's patentable and what's not. And many of those methods of doing business were invalidated. And I think same thing's going to happen with these gene patents. You should not be allowed to patent a gene itself. You should be allowed to patent the test for that gene. And USPTO has sort of, in my opinion, has overstepped its bounds a little bit. I do have a background in patent law as well. That's why I'm offering that.
Take one last question.
One last question. Yes. I think the general knowledge and the general, would current platforms, engineering software platforms, be able to do the work that we want to do? Yes and no. I think the fundamentals are there. The fundamentals of what is needed in life sciences is there. But obviously the parameters have to change. You know, we have to go from mechanical things to like system biology level things. But what is needed is there. What service is being offered is there, and the understanding. And to me, actually more important than that, because someone can figure out the technology. It's really the business mindset is there.